Why does a revolutionary cancer drug melt away tumors in one patient, but, completely fail in the person sitting in the very next hospital bed?
Yeah, it's really the million-dollar question in oncology right now.
Right, because you can have two people with identical genetics. They're battling the exact same type of melanoma, receiving the exact same IV infusion. But for one, it's a total miracle cure.
Exactly.
And for the other, the tumor just keeps growing as if the drug, isn't even there.
It is, it's honestly one of the most agonizing mysteries in modern medicine. We really want medicine to be this simple input-output equation, right?
Oh, absolutely.
But when you look at these extreme variations in patient outcomes, it forces you to realize we are missing a massive variable in that equation. And the answer to why that happens, well, it isn't found in the tumor itself.
No.
Or even in the patient's human DNA.
It's sitting right in their gut, which completely blew my mind when I was going through the research for today's deep dive.
It's a huge paradigm shift.
It really is. So we are looking at this incredibly comprehensive 2026 review from the journal Cell Metabolism by Gladstone and Sonnenberg.
Great paper.
Yeah, and they map out what they call the gut organ axis. And the core idea here is that the trillions of microbes living in your gastrointestinal tract aren't just, hanging out digesting your lunch.
Right. They're definitely not just a passive processing tube.
They are an active, highly sophisticated control center for your entire systemic immune system.
Historically, medicine treated the human body as this isolated, sterile system. But this research fundamentally reframes human biology. Those microbes are constantly communicating with, and in many cases actually directing, the immune cells that patrol your entire body.
So to understand how they dictate the success of our most advanced cancer treatments, we first really need to unpack how they interact with cancer directly.
Exactly, because they are not neutral bystanders. Some actively drive the disease.
Let's look at the dark side first, then. The review points out that specific strains of Escherichia coli, or E. coli, actually promote tumor growth. And they do this by producing something called colibactin.
Right, colibactin. So colibactin is what we call a genotoxin.
Meaning it's toxic to genes.
Exactly. Its sole function in this context is to damage human DNA. It physically alkylates the DNA.
Okay, let's break that down. Alkylation. What does that actually look like inside the cell?
Sure. So imagine your DNA as a zipper. Alkylating it means the bacteria is attaching a rogue chemical tag directly to the teeth of that zipper.
Oh, wow.
Yeah, so when the human cell naturally tries to unzip its DNA to replicate, that chemical tag causes structural chaos. The zipper gets stuck, the DNA physically breaks, the cell basically panics, and it leads to rampant genetic instability and mutations.
Which is the exact recipe for cancer.
Precisely. And it doesn't just stop at direct DNA sabotage either. Other bacteria produce virulence factors that trick your immune system.
Into doing what?
Into chronically producing an inflammatory signal called IL-23, which then triggers another cascade, resulting in a molecule called IL-17.
So it's basically setting off a false alarm that never stops ringing.
Yes. And the end result is this state of localized chronic inflammation.
Which is exactly what tumors want.
Right.
Because chronic inflammation brings a constant supply of blood, nutrients, and growth factors to the area.
It does. The microbes are essentially terraforming the local environment. They're making it a perfect nursery for cancer cells to thrive.
It's like a microscopic demolition crew paving the way for the tumor. But on the other side of the battlefield, we have microbes acting as a defense force.
Right. It's not all bad news.
Yeah, like Lactobacillus reuteri, for example. The source says it produces a molecule called reuteri.
Reuteri is fascinating. It acts almost like a localized molecular bleach.
A bleach.
Yeah. It increases what we call reactive oxygen species, or ROS. These are highly volatile oxygen molecules that severely restrict the growth of colorectal cancer cells. They basically poison the tumor environment.
So you have a demolition crew of rogue bacteria destabilizing your cells, and this defense force actively trying to bleach the tumor. But here's where the research took a turn I absolutely did not expect.
The intratumoral microbes?
Yes. It's one thing for microbes to fight over territory in the colon, but the review highlights that microbes have been found physically living inside tumors across various cancer types: melanoma, lung cancer, breast cancer.
It's wild.
Wait a minute, though. If these microbes live in the intestines, how on earth are they getting inside sterile tumors in completely different parts of the body?
Yeah, this is one of the most fiercely debated topics in the field right now. Like, how do they cross that distance? The prevailing theory really comes down to oxygen.
Okay, how so?
Well, Both the deep layers of your gut and the dense, chaotic center of a rapidly growing tumor share what we call a hypoxic state, meaning they are severely starved of oxygen.
Right, because tumors grow so fast that they outpace their own blood supply, right?
Yeah.
And the blood vessels they do manage to build are, like, chaotic and leaky.
Exactly. They create these oxygen-deprived dead zones in the middle of the tumor. So the theory is that certain gut microbes manage to escape the intestines, perhaps by hitchhiking inside immune cells.
Wait, hitchhiking?
Yeah, using immune cells like macrophages as Trojan horses. They survive inside them, travel through the bloodstream, and when that macrophage wanders into a hypoxic tumor pocket, the bacteria realize it's a perfectly welcoming, oxygen-free home.
It mimics the deep gut.
Ah,
That is deeply unsettling.
that is a little creepy, yeah.
But incredibly fascinating. Okay, before we move on to the actual drugs, let's do a quick bullet recap for you listening just to lock this in.
Good idea.
Bullet 1. Microbes can cause cancer by mutating DNA with colibactin or driving inflammation via IL-17. Bullet 2. They can fight cancer using localized bleach like Redorin. Bullet 3. They might actually travel and hide inside the oxygen-deprived centers of tumors.
Perfect summary.
So a question for you listening. Knowing there's a microscopic war in your body right now, which side is your microbiome on?
It's a great question because that balance of power becomes literally a matter of life and death when a patient starts immune checkpoint blockade therapy, or ICB.
ICB.
Okay.
Okay, let's get into the main event. Since we know microbes naturally fight or fuel tumors, how do they interact with their most advanced medical weapons?
Well, ICB is arguably the greatest revolution in oncology in the last few decades. But to understand why it's so powerful, you have to look at how cancer actually survives the immune system.
Right, it hides.
Right. Your immune system has these highly lethal assassins called cytotoxic CD8 T cells. They are specifically designed to hunt down and kill mutated cells. But to prevent those assassins from accidentally attacking your healthy tissue and causing autoimmune disease, they have built-in off switches.
These are the checkpoints like PD-1 or CTLA-4.
Exactly. And the tumors exploit that. They evolve to physically press those off switches. It's like the tumor puts on an invisibility cloak, so the T cells just float right past, completely ignoring the cancer.
And so ICB drugs are designed to block those off switches. They rip off the invisibility cloak, taking the brakes off the T cells.
Yes. But here is the absolute bombshell of the review. Taking the brakes off isn't enough.
What do you mean?
If you give a cancer patient broad-spectrum antibiotics alongside their ICB therapy, the cancer drug's efficacy completely plummets.
Oh, wow.
Yeah. Clinical studies show drastically shorter progression-free survival in cancers like melanoma and lung cancer when antibiotics wipe out the patient's gut flora.
That fundamentally changes how we view these drugs. I mean, I initially thought of ICB like a sophisticated facial recognition system. The drug turns the security cameras back on so the immune system can see the tumor.
Right.
But what this research is actually saying is that the gut microbes provide the database of the criminals' faces. You can turn the cameras on all you want, but if the antibiotics have deleted the database, the immune cells still don't know who to attack.
That captures the dynamic perfectly. The T cells require these secondary priming signals that only specific healthy microbes provide.
And the researchers identified the specific heavy hitters, right? The good bugs.
They did. Bugs like Akkermansia mucinifila, various bacteroid species, and bifidobacterium.
And what do they actually do?
When these specific bugs are thriving in the gut, they increase the systemic production of crucial immune signals like IL-12 and interferon gamma. They literally arm the T cells for battle.
And the clinical proof of this is staggering. We have to talk about the fecal microbiota transplant trials, FMT.
Yes, the FMT trials are incredible.
They took patients whose tumors were completely ignoring the ICB drugs, patients who were essentially out of options, right?
Right.
And they gave them a fecal transplant from a patient who had survived their cancer thanks to the same drug.
And it literally transferred the cure.
It was just...
I know. By transplanting the microbial ecosystem, the non-responder's immune system suddenly woke up, recognized the tumor, and began attacking.
They even found that fecal matter from a healthy donor, like someone who never even had cancer, could overcome ICB resistance just by restoring that baseline microbial diversity.
It's really profound.
Which brings up a really counterintuitive point from the paper. If someone wants to boost their gut bacteria, say they get a cancer diagnosis, they might just run to the pharmacy and buy a commercial probiotic.
Which you would think is a good idea.
But the paper notes that taking over-the-counter probiotics can actually reduce a patient's responsiveness to cancer therapy. That seems completely backwards. Aren't probiotics supposed to be the good guys?
It does seem backwards until you look at the gut as a complex rainforest ecosystem. A healthy microbiome derives its power from immense, intricate diversity, thousands of species keeping each other in check.
Okay.
When you swallow a commercial probiotic, you are dumping billions of copies of just one or two bacterial strains into that delicate ecosystem.
So they act like an invasive species.
Exactly. They can temporarily overrun the gut, outcompeting the natural, diverse network of microbes like Akkermansia that the immune system actually needs to communicate with. It simplifies the ecosystem, which weakens the immune response.
Okay, let's do our second mini recap. Bullet 1. ICB works by taking the brakes off immune T cells. Bullet 2. Taking antibiotics destroys the database the T cells need, ruining the drug's efficacy. Bullet 3. Fecal transplants can literally transfer a cancer cure by restoring the ecosystem.
Spot on.
So, question for the listener. If your life depended on it, would you accept a fecal transplant to make your cancer therapy work? It's intense to think about.
It really is.
But it leads us to the next massive question. If the tumor is in the lung and the crucial microbes are down in the gut, how exactly does the immune system bridge that physical distance?
Right, the long-distance communication. The paper uses a very helpful framework to explain this, the TMM pathways.
Translocation, mimicry, and metabolites. Let's start with T translocation.
Translocation is the physical movement of the bacteria. The review notes that administering an ICB drug actually causes specific gut microbes to cross the intestinal barrier and travel into the mesenteric lymph node.
And what do they do once they get there?
They act as an immunologic adjuvant. They set off the alarm bells, priming and training dendritic cells, which then leave the node and direct the systemic immune response.
Wait, I need to stop you there. Bacteria escaping the intestinal barrier. Doesn't that just mean the patient has a leaky gut? I thought systemic leakage was incredibly dangerous.
It is.
So why is it suddenly a superpower here?
It's a vital distinction. Uncontrolled, chronic, leaky gut, where large amounts of bacteria flood the bloodstream, is extremely dangerous. It causes sepsis. But what we are talking about here is highly localized, controlled sampling.
Controlled sampling.
Yes. The immune system is intentionally lowering the drawbridge just enough to allow a few microbes into the lymph node. It uses them as a localized stimulant to train the immune cells without letting the infection spread to the blood.
Okay. A controlled burn instead of a forest fire. Got it. Now, the first M, mimicry, or antigenic cross-reactivity, this mechanism is pure biological espionage.
It really is. The review explains that certain microbial proteins look incredibly similar on a molecular level to tumor proteins.
They basically share a disguise.
Which leads to a highly effective case of mistaken identity. The immune system in the gut encounters these specific bacteria and trains what we call T helper 17 cells, or Th17 cells, to attack them.
Okay.
These Th17 cells mature, eventually transition into Th1-like cells, travel throughout the body, and eventually bump into the distant tumor.
And because the tumor is wearing a molecular disguise, that looks just like the gut bacteria.
Exactly. The immune cells unleash their entire arsenal on the cancer. They think they're fighting a localized bacterial threat, but they end up destroying the cancer entirely by accident.
That is just brilliant. And that brings us to the final M, which is metabolites. These are the chemical messengers the microbes produce that enter the bloodstream.
Yes, and this is where things get really actionable.
Right, because these metabolites are heavily influenced by our daily choices. Before we dive into that chemical arsenal, recap time. TMM travel, or translocation to the lymph nodes. Mimicry, where immune cells confuse tumors for bacteria. And metabolites, the chemical signals.
Good memory device.
So can you, listening right now, picture your gut actively training immune assassins through molecular mimicry? It's wild. Okay, let's zoom in on these metabolites.
We really need to. When microbes digest our food, they excrete byproducts. For a long time, we thought these were just waste.
No, they're not.
No, they are incredibly potent biological signals. Let's take short-chain fatty acids, or SCFAs, like butyrate and pentanoate.
The heavy hitters of the gut world. The paper details how SCFAs physically enter the CD8 T cells and crank up the function of a cellular metabolism sensor called MTOR.
Right, MTOR. What is MTOR? Let's unpack that. It's essentially the cell's metabolic engine. It senses nutrient levels and tells the cell whether to grow, divide, or conserve energy.
So by boosting MTOR, the microbes are directly injecting high-octane fuel into the immune system's assassins.
Exactly. Boosting their tumor-killing power while simultaneously shutting down immunosuppressive enzymes.
Okay, but I have a pushback question about this. If SCFAs like butyrate are so amazing, why did the source mention an experiment where scientists put SCFAs directly into the drinking water of mice and it actually hindered their anti-tumor immunity?
Yeah, that surprised a lot of people.
Right. Why would giving them more of a good thing ruin the immune response?
Because biological context is everything. It's the paradox of location.
Okay, explain that.
When SCFAs are produced naturally in the colon by fiber-fermenting microbes, they interact with the immune cells in that specific, highly localized lymph node environment.
The controlled burn we talked about earlier.
Right. That local interaction creates the perfect stem-like T-cell response that travels to the tumor. But when you bypass the gut ecosystem entirely and flood the systemic bloodstream with SCFAs via drinking water,
It confuses the system.
it creates a global immunosuppressive effect. You can't just isolate a single chemical from the ecosystem, dump it everywhere, and expect the same targeted result.
It's not just what you deliver, it's exactly where and how it's delivered by the microbes.
Okay, what other metabolites are in this arsenal?
Well, you have compounds like inosine, produced by specific strains of bifidobacterium. Inosine physically binds to receptors on T cells to sustain their energy during prolonged fights with a tumor.
Oh, and the exercise link. We have to mention formate.
Yes, formate enhances T cell effects via NRF2 signaling. But the fascinating part is that formate production by the microbiome is induced by physical exercise.
So the literal mechanical stress and metabolic demands of working out change the chemical output of your gut to favor tumor-fighting compounds.
It does. We also have indoles, specifically indole 3-propionic acid, or IPA. This one is amazing because it alters how DNA is packaged inside the immune cells.
Okay, wait, how does it do that?
It acts on the physical architecture of the T cell's genetics. DNA is usually spooled up incredibly tight around proteins, like yarn on a spool.
True.
That tight packaging restricts which genes can be read. IPA alters a process called H3K27 acetylation.
That sounds like a password. It basically acts like a chemical key that unlocks and unspools specific sections of that DNA, granting the cell access to stem-like programming.
Meaning the T cell doesn't just attack once and then die of exhaustion. It becomes a resilient, self-renewing cell that can keep fighting a relentless tumor without burning out.
Exactly. And then there are the secondary bile acids. The specific mechanism here is incredible.
Let's hear it.
There's a specific gut chemical 3-oxodelta 406 LCA.
Rolls right off the tongue.
Yeah, quite a name. But this chemical enters the blood and physically acts as an antagonist to the host's androgen receptor.
Meaning, if a patient has a tumor that relies on androgen signaling as fuel to grow, this gut chemical physically blocks the fuel pump.
Right. It starves the tumor from a completely different angle. It is a vast, sophisticated pharmacy operating inside us.
Okay, quick recap on the chemical arsenal for the listener. Bullet 1. SCFAs boost T-cell engines, but location matters. Bullet 2. Exercise literally induces tumor-fighting formate. Bullet 3. Bile acids can physically block a tumor's fuel receptors. So are you feeding your internal chemical factory the right raw materials to produce this arsenal? Because that brings us to the most practical aspect of this deep dive.
Diet. Diet is huge here.
We are the ones feeding this factory. The review specifically contrasts the impact of a high-fiber diet with a high-fat or ketogenic diet on cancer outcomes.
And the differences are stark. High-fiber diets provide the raw, complex carbohydrates that these beneficial microbes need to ferment,
which fuels the production of those SCFAs we just discussed.
Exactly. The data shows that high-fiber intake significantly improves survival rates for patients on ICB therapy, specifically because fiber creates these inulin gels in the gut that facilitate the creation of those durable stem-like T cells.
Conversely, a high-fat or ketogenic diet lowers the abundance of crucial bugs like bifidobacterium and the T817 cells responsible for that molecular mimicry we talked about.
Now, to be fair, keto can have some very complex, occasionally beneficial metabolic effects for certain other conditions. But in the specific context of ICB therapy, it generally deprives the microbiome of the raw materials it needs to build that diverse, cancer-fighting chemical arsenal.
Right. It's about the specific therapy context. But we have to address the danger of revving up the immune system this intensely. There is a massive catch.
There always is. The source calls them IRAEs, immune-related adverse events.
Because when you permanently take the brakes off the immune system to hunt a tumor, sometimes it doesn't stop there.
It starts attacking healthy tissue.
Right. Patients can develop severe, debilitating autoimmune-like side effects, such as violent inflammation of the colon, known as colitis.
It is honestly the biggest limiting factor for these life-saving drugs, but the twist in the research is that the microbiome dictates this toxicity too.
Let's unpack that. How?
A specific bug called Bacteroides intestinalis was found to upregulate a protein, IL-1 beta, that drives the severe gut inflammation.
But the truly mind-blowing part is that the bacteria driving the side effects are completely different species from the bacteria driving the cure.
Yes. For instance, bacteria that produce a specific metabolite called menaquinone might actually suppress that inflammation and prevent the toxicity without stopping the antitumoral attack.
So wait, if the therapeutic bugs and the toxic bugs are distinct species, does that mean we could eventually design a custom probiotic?
Yeah.
Like one that gives us 100% of the cancer cure with 0% of the side effects?
That is the ultimate holy grail of this field. If we can map exactly which species drive the cure and which species drive the debilitating inflammation, we could engineer bespoke, highly targeted microbial treatments.
That would change the landscape of oncology forever.
Absolutely.
Time for our penultimate recap. Bullet 1, fiber fuels T-cell stemness. Bullet 2, hyperimmunity causes severe side effects, or IREs. Bullet 3, specific distinct bugs dictate both the cure and the toxicity.
Nailed it.
So how might your breakfast today be writing the code for your immune system's behavior tomorrow? Something to think about.
Definitely. But just when you think you have a handle on how this all works, how the gut microbiome acts as a switch to make the drug work, Gladstone and Sonnenberg drop one final radical paradigm shift.
The gut-first paradigm.
Right. Up until now, the assumption has been that the ICB drug enters the blood, goes straight to the tumor, and takes the brakes off the T cells there, while the gut microbes just send supportive signals from afar.
But this new theory proposes that the drug actually alters the gut first.
It's a complete inversion of how we thought these drugs worked.
Okay, break this down for us.
The researchers found that there are these unique immune cells permanently stationed in the intestines called unconventional lymphocytes, specifically ILC3s.
Okay.
And these gut resident cells naturally express the exact same checkpoint molecules like PD-1 and CTLA-4 that the cancer drugs target.
So when a patient receives an IV infusion of a systemic cancer drug, a significant portion of it goes directly to the gut and binds to these ILC3s.
Exactly. Now, one of the main jobs of an ILC3 is to produce a cytokine called IL-22, which is absolutely crucial for maintaining the structural integrity of the gut barrier. It keeps the wall strong.
But when the cancer drug blocks the checkpoints on those ILC3s, it forces them to reduce their production of IL-22.
And without enough IL-22, the gut's barrier integrity drops. It becomes slightly more permeable.
And this is the brilliant, slightly terrifying mechanism. This intentional drug-induced drop in barrier integrity might be exactly what provokes the microbial translocation.
The controlled sampling in the lymph nodes we discussed earlier.
So it's an inside job.
Basically, yeah.
The cancer drug isn't just floating over to the tumor site. It is intentionally breaking down the gut's walls just a tiny fraction of a millimeter to let the microbiome out.
To sound the alarm and train the systemic immune response, it suggests that the mild side effects in the gut aren't just collateral damage. They might be the foundational mechanism of action that makes immunotherapy work in the first place.
The drug triggers the gut trains the immune system, and the immune system kills the tumor.
It completely upends how we view pharmacology in the human body.
Let's do our final synthesis. We've gone from looking at the gut as a simple digestion tube to realizing it's the command center for our body's defense systems.
We really have.
We've seen how microbial warfare involves DNA-damaging colibactin and tumor-fighting redorin, how the mechanisms of translocation, mimicry, and metabolites allow gut bacteria to direct targeted strikes in the lungs or the skin,
Right.
how diet shapes that chemical arsenal, and how the cancer drugs themselves might be relying on the gut to launch the attack through this mind-bending gut-first paradigm.
It's a lot to take in, but considering the sheer scale and complexity of what is happening inside you right now, it leaves us with a profound philosophical question to grapple with.
What's that?
Historically, we have always defined the immune system in very simple, binary terms. Self fighting non-self. Human cells destroying rogue human cells.
Right.
But if our ability to survive cancer relies entirely on the genetics, the chemical arsenals, and the physical pathways of trillions of alien microbes living inside us, well, when the tumor finally melts away, who exactly is doing the curing, us or them?